hopefully it's working now. All right, hopefully this is working now. So Bio 151, this is Dr. Granier. Today's Wednesday, it's 10:30, and we have the biology of human health and disease going on, week 3 over the cardiovascular, the immune, the respiratory, digestive, and muscular organ systems and pathologies. Your book went over in chapter 12 and 13 some of the cardiovascular and some immunity, but your book was insufficient on meeting the requirements set forth by our syllabus concerning the respiratory, digestive, and muscular organ systems. So while today or this morning I'll try to do the cardiovascular and immune systems, there's a second set of slides and a second kind of plan for this afternoon for review of those organ systems and those pathologies related to the respiratory, digestive, and muscular system. I noticed on worksheet 4 that I completely forgot to do muscular organ system pathologies. So please be aware that that was a mess up on my part. You are still required to look at the muscle system and pathologies associated with your muscle organ system, your skeletal muscle system. Okay. So again, our objectives really are to go through the anatomy and the physiology of each of these organ systems and then try to hit on 1 to 2 of the common pathologies per organ system that you need to be familiar with. I always have a few extra listed because we need to be able to translate. Because of this deficiency at the cellular, tissue, or organ level, that leads to this functional problem for the organism related to this organ system. All right, so I started with the immune system. The immune system in some of your anatomy and physiology textbooks is also known as your lymphatic system. It's composed of lymphatic vessels, lymphatic organs, which are the tonsils, the spleen, these like Peyer's patches, these malts, these little areas in your GI tract that sit right behind your epithelial tissue. And this is a second-tier defense system behind your epithelial barriers in the immune system's nonspecific defenses. And so when you talk about the immune system, you're talking about usually all the white blood cells and all together the mechanisms to include the skin, to include the lymphatic system, to include lymph nodules, to include the spleen, plus all the white blood cells that help us defend against disease-causing microorganisms or toxins or allergens or whatever that would cause damage or destruction to our cells and tissues. Okay, so these systems get lumped together. The lymph system is a little bit different from the immune system. The lymph system, in some ways, it's a way to return exogenous interstitial fluid, so extra fluid that would accumulate around cells. It helps us return that fluid to the blood and the circulatory system. So it's a network of, as the picture over here shows you, it's a network of tubes, all right, that collect fluid and bring that fluid back to the heart, okay? Back to the heart, back into venous blood supply. If something happens and that fluid's not returned, then the limb would swell and you would get edema. And you see that with like breast people when they have like the breast, the mastectomy, and all of that lymphatic tissue. Sometimes they have problems with swelling in the arm of the side of the breast that they had the surgery. And so it's because they lose all this lymphatic system. Now, while we're returning all this fluid to the blood supply, and it takes a little bit more time because it has to go through a series of vessels that are fighting gravity, and they're entering these nodules, we're going to do a filter, and we're going to do a check in this fluid and a check in this lymphatic juice to make sure that we don't have any pathogens that have made their way through our organ systems. So the immune system is tied to the lymphatic system in that much of what the organs of the lymphatic system are, these lymph nodes and spleen and tonsils, are these big rich sites of white blood cells that are there to be a barrier behind and be a defense against behind the epithelial barriers, and then to also do a job of filtering and removing dead debris, proteins, ions, things that shouldn't be in our interstitial fluid, and that can include viruses, bacteria, and any kind of pathogen. Okay. So it's important to learn about the components. And if in this picture you can see the spleen. This is just representing GI-related aggregation of white blood cells. So it's kind of like an area behind the small intestines, the MALT, it's known as. You have prior patches in there. And then all of these little connection points, those are lymph nodes. Okay. The thymus gland is where T cells of white blood cells mature, and then your bone marrow will sometimes get looped in here because that's where your other white blood cells come from and mature. All right? So the white blood cells are going to be more a part of the immune side, and any antibodies and proteins that they make are also usually tied to that. So the proteins complement the interleukins, the cytokines, the interferons, all of those proteins that can play a role in killing and destroying any cells or pathogens in the body. All right, so the takeaway is there are quite a few different white blood cells, but they all come from the same spot. In your red bone marrow, there is the stem cell, and it's going to give rise to two pathways, either the myeloid pathway or the lymphoid pathway. In the lymphoid pathway, you give rise to B cells, T cells, and natural killer cells. T cells play a key role, and we learned this with AIDS, in being able to transition from non-specific immune defenses to specific defenses. So T cells are critical to turn on B cells and make them produce specific antibodies towards a specific pathogen, as well as to turn on other specific T cells that can target and kill cells that we know don't belong to ourselves or have been invaded and overcome by virus and bacterial pathogens. Okay. On the myeloid side, yes, we can get red blood cells and platelets, and that's important in the blood. But from the immune standpoint, we give rise to what's known as these granulocytes, and it's not mentioned here, but also a monocyte, which is known as a macrophage. So a monocyte and a macrophage. Okay, and so all of the other white blood cells, based upon the influence of, is there EPO, which would lead to red blood cell production, erythropoietin. Is there TPO, thrombopoietin, that would give rise to platelets. Lack of those things and exposure to other cytokines and other immune type related proteins are going to give rise to all of our cells, the white blood cells, either our granulocytes, because they have all these dense granules full of materials that the cell can then spit and release and maybe break holes because it can damage, it's toxic to other viruses and bacteria, or parasites, or the monocyte, which is going to be a big cell that's capable of eating any foreign object. Okay? So again, just a different way to see what are the types of lymphocytes. So there are more than just T cells, B cells, natural killer cells. There are subtypes. So the T cells are the big category. And again, they get the name T cells because they are going to leave red bone marrow at some point, head to the thymus gland, all right? And in the thymus gland, they are going to become competent. And part of these cells becoming competent means they have the ability to understand if something is self or not self, right? And the key cells that help become competent in determining self and non-self are these helper T cells. And they are the link that ties non-specific immunity to specific immunity. Right? The rest of these T cells, cytotoxic T cells, play a role in killing other cells. And they can kill any cell that would be foreign. So a T cell recognizes it as not self. They can kill any cell that's been, surrounded by bacteria or surrounded by antibodies, so we know it's infested with virus or bacteria. And then it can infect potentially any cell that's just acting really funny and starting to express antigens that are inappropriate, proteins on their membrane that are not correct. Right? Your suppressor T cells are somewhat your brakes. They try to keep the cytotoxic T cells from overdoing things and killing healthy tissue. And your memory T cells stay around and remain in our system somewhat long term or for an extended period of time. So if that pathogen, that same bacteria or virus or parasite ever enters again, you already have the specific mechanisms ready to ramp up and defeat it. Right? Now, B cells get their name because they are going to mature in bone marrow. And bone marrow cells, so they never leave the bone marrow until they're ready to start circulating. And most of the cells that leave the bone marrow are called naive. So they're not quite ready to make specific antibodies towards a specific pathogen. So they make really big global non-specific antigens, or antibodies, sorry, against lots of antigens. All right? Now, eventually though, if the T helper cells get exposed to something that we know is foreign, part of what they can do is interact with these naive B cells and trigger them to undergo a change where instead of making kind of global antibodies against lots of different proteins, they can help introduce an antigen that we know is not self, that we know is maybe associated with the chickenpox virus, or maybe it's associated with the tuberculosis bacteria. And so now that B cell, upon exposure to that antigen, and knowing that it's not self, can rearrange its genetics and its gene expression when it makes its antibodies to make antibodies that specifically find and target that corresponding antigen protein. And when it does that and it makes a plethora of antibodies, it's now a plasma cell, right? And every once in a while, again, some of these B cells in the division process of becoming a plasma cell will actually, some of them will stay and become a memory cell. And by staying and becoming a memory cell, like our T cells, they retain that ability to make specific antibodies against a specific antigen for the lifetime of that memory B cell's presence in your system. So that's why we, upon exposure to chickenpox, we never have to worry about getting chickenpox again. Or upon exposure to certain diseases, you should never have to see them again. So if you ever had to have smallpox, you know, it should never happen to you again, okay? Because you have those memory cells. Now the natural killer cells, at one point, somebody thought they were actually a form of cytotoxic cells. And so this is actually kind of new in the immunology world. And what these cells do is they're part of non-specific, okay, immunity. And so their goal is to survey and keep an eye on all of your cells in your body to make sure that they don't become cancerous. So they are what is constantly circulating in your interstitial fluid, rubbing up against cells, and ensuring that the cell is acting according to the normal cell cycle and the normal antigen expression. So these should be the cells that if little cancer cells start to, or cells turn cancerous and start acting inappropriate and start undergoing lots of mitosis inappropriately, they would be the ones to try to counteract that. Okay? And so cancer then arises in your body when these cells are not finding and destroying those cancerous cells. Right? Okay, so I've mentioned some of these defenses. So your body has kind of two tiers of defense. It's got kind of the baseline defenses that are always in play against whatever is thrown at it. Your skin. Your skin is made up of epithelial tissue, that is cell-touching cell, making an impenetrable barrier against something getting into underlying tissue and into your bloodstream. And that is a non-specific defense. And that is also the first line of defense in many cases. Right? The mucous membranes of your nose and your respiratory passages. Those are another first line of defense. They're epithelial cells. They make a sticky mucus. You try to trap anything that would get in through the airway that way and capture it and spit it out, put it in the stomach, whatever. And then you have other mucous membranes like in the vagina, around the urethra, that again is trying to make a clog and a plug so things don't get up in the passageways and the breaks in the epithelial tissue of your skin in other areas of your body. Right? And it doesn't matter if it's a fungus, it doesn't matter if it's a worm, it doesn't matter if it's a virus, it doesn't matter if it's a bacteria. The skin, the mucus, it's all about not getting it through And if it does get near or touching, stick to the mucus, stick to the secretions, and get washed off and flushed away. All right? Now, the other nonspecific defenses are right behind the skin and the mucus. This is your white blood cells and some of your cells that take up residence in your dermis of your skin, so the underlying connective tissue behind the skin. And their goal is, if, let's say, you have a break or a cut, they can then, if something or a virus or a parasite or a worm tries to get through this epithelial lining, they are then hopefully there to stop it. And they can stop it through a variety of mechanisms. One of the things they can do is eat it, phagocytose it. So they endocytose it, they eat it, they take it inside themselves, and in taking it inside their cellular wall, they can then digest it, break it down, and not let it then get in your bloodstream. They can make proteins that that can maybe destroy it. So those granulocytes have things like histamine, have things like nitric oxide, have different proteins and different molecules that could potentially punch holes in a bacterial cell, punch holes in a worm, punch holes in a parasite, make a tick unable to live in that environment. And by doing that, could kill it, right? And then the other thing that happens is some of those molecules that are released by those white blood cells or those cells in that local area, so the connective tissue cells or even the epithelial cells, is going to be to recruit inflammatory type mechanisms. So recruit more white blood cells to the area, recruit and change blood flow dynamics, recruit and change water retention dynamics. And in doing so, you get an inflammatory response. Right? Now, ultimately, some things are very crafty and they get past these non-specific defenses, and so they then try to take up residence inside your body and propagate and survive. And so it takes a little bit more specific defending against that pathogen to then remove it without removing healthy, good tissue. And this is where our specific defenses come into play. So should something get past our first tier and second tier of defenses, should something survive and continue to propagate and take over areas of the body or cells, this is where our antibodies and our specific defenses come into play. And their first goal is they have to find that antigen's presence, that pathogen's presence. And in this phagocytic process of step 2, that is one of the ways they get found, because these cells that eat a foreign object present it to T helper cells. And it's these T helper cells, these lymphocytes, that say, yes, you ate something foreign, or no, you ate dead epithelial tissue, dead cells that belong here, and will trigger, potentially, if it is a foreign object, the rise of antibodies and B cells turning into plasma cells, and the rise of cytotoxic T cells that can go and find cells that have been overcome by virus or bacteria and start to destroy them. Okay? So these defenses are always in play. These defenses take time. So this is why when you get exposure to the common cold may take 7 to 10 days to run its course. That means it takes 7 to 10 days for these specific defenses to get ramped up enough to start overcoming the virus that has invaded your body. Right? It takes 7 to 10 days for these non-specific defenses to finally realize they've been penetrated and find some of that virus to eat, and then it present to the T helper cell and get the T helper cell to initiate a specific defense. Okay? Whereas if we know what sometimes we're sick against, like we know it's strep throat, and we don't want to wait 7 to 10 days for our specific defenses to come into play, which they should, we sometimes can give people antibiotics. So we can give antibiotics early in the exposure to kind of assist along the destroying of the bacteria or the bacterial infested tissue. Okay? So again, when you look at specific defenses, all right, when you activate the T helper cell, it's going to initiate 2 cascades of defense. It's going to initiate 2 cascades of pathways towards overcoming that virus, bacteria, fungus, parasite. The first is going to be a protein-driven defense, and that's going to involve the B cells, and that's going to involve the B cells making antibodies. Antibodies are little proteins, and they're shown here by the little Y's, and what they have is this area on each protein that specifically finds the bad guy, the pathogen, and the protein expressions on the pathogen. And by binding to that pathogen and binding to those pieces and parts of the pathogen, it helps keep them from hiding. It helps keep them from maybe going unnoticed in your body. It makes a bigger target, a bigger molecule with a big red flag on it. And so then that molecule can be taken up by phagocytic cells, such as your monocytes or your macrophages or your neutrophils, and then taken in, destroyed, and removed from your body, and removed from causing damage. And in order to get this humoral immunity and these B lymphocytes activated, the T helper cell has to initiate that. Okay, and it initiates it because it gets exposed to the antigen, because a macrophage, that second line of defense, that neutrophil, that second line of defense, ate it, showed the T helper cell, this is what I ate. The T helper says, yes, you ate something foreign, and makes sure, and then turns on the B cells. The other side of this equation is the T helper cell gets exposed to the antigen, sees that it's not self, and will then initiate cellular immunity. It initiates these, here's the T helper cell here, it initiates these cytotoxic T cells to make cytokines that can then target cells that have been invaded and then destroy those cells that have been invaded by an antigen or a pathogen that doesn't belong. Okay? All right. So again, other times you'll see specific immunity is also called adaptive immunity. And adaptive immunity, making ourselves make antibodies, make ourselves have memory cells towards a specific pathogen, can be acquired through two mechanisms. It can be acquired naturally because we naturally catch the flu and therefore have the flu in our system for 7 to 2 weeks and naturally go through that process of making our own antibodies against it. Or we can naturally be a baby and drink mom's breast milk and get mom's defenses because mom's been exposed to those antigens already. So those are our natural ways to acquire specific immunity, against a pathogen. We either naturally get sick and overcome, so you get chickenpox and you overcome and you never have chickenpox again in your life, or you're a baby and you get it because you're in mom, or you drink mom's breast milk. Okay? Now, of course, it can take 7, 10 days, 2 weeks, and we don't always want to wait that long for our body to overcome a pathogen, especially pathogens that could kill you in 2 or 3 days, because you're dead before your system can overcome it. So there are artificial ways we've learned to overcome pathogens and help the immune system along. And to artificially acquire specific immunities against a specific pathogen, we can do that in an active way, such as giving exposure to non-virulent pathogens. So that's where vaccines come into play. We give you a piece of your smallpox vaccine, or a piece of your chickenpox vaccine, or a piece of the flu virus, and we let the body respond to that non-virulent, non-alive strand of virus or bacteria, generate antibodies against it, so if you ever are exposed to the real virus or the real protein or the real bacteria, you can get through overcoming it faster because you already have some of the specific defenses in place. Now, the way to passively do it would be to give someone IV antibiotics, to give someone some type of, okay, I know you are suffering from the E. coli virus. So we know that someone just overcame the E. coli virus, so we're going to give you a bag of their blood because their blood has antibodies already against this outbreak of E. coli, and they overcame. So that would be one way. Or we're going to give you IV antibiotics, full of antibiotics against a specific pathogen, right? So adaptive is just another way to say specific immunity, and you can acquire specific immunity naturally by getting sick or being a baby and getting it from mom. You can get specific or adaptive immunity artificially by getting exposed by vaccines, getting exposed to less virulent forms, but allowing your B cells and your T cells to undergo a natural process, even though they are being exposed to a non-deadly or a non-sick-making form of the pathogen, but you still go through that normal activation of the T cells, activation of the B cells, and make your own antibodies. And then the most lazy way is, of course, to just know, okay, I'm going to take the antibodies already that are pre-made against this pathogen. I'm just going to take them by injection or take them by IV blood or whatever. And so that way I will never have to make my own because I'll just defeat it with passive mechanisms without my system ever making memory cells and ever making their own antibodies. Okay. All right, so let's talk about some of the pathologies. So lymphedema is any abnormal condition where the lymphatic vessels, the lymphatic organs, do not do a good job of returning fluid to the venous or cardiovascular system. And so anything that leads to swelling or edema in the interstitial fluid around your cells is a lymphedema pathology. And you can get it because vessels get blocked. So there's some worms out there that can block vessels, a parasitic worm. And when it happens, we can call it specifically elephantitis. And go ahead and Google elephantitis. Just prepare yourself. The pictures are kind of gross, all right? So the worm, the parasite, blocks so effectively that there's so much fluid and so much edema in the area around the cells of the arm or the leg that the limb swells and expands and looks like an elephant-sized leg or an elephant-sized arm. It's really gross, all right? But this happens post-surgeries. Like I said, especially in mastectomies, this is a big deal of they take the breast tissue, they take some of the lymph nodes to test to see if the breast cancer spread. And the problem is by taking those lymph nodes, you're taking away the tubing that puts extra fluid from the right arm and the left arm back into your cardiovascular circulation. So without that tubing, you now have excess fluid staying in the arms, and it takes longer for it to naturally just osmotically diffuse in. So it might lead to lymphedema in the right or the left arm, depending, or both, if it's both breast and lymph nodes removed. And this is a big surgical complication in breast tissue removal. Okay. And so really the other type of pathology could be anytime, again, some of the cells in lymphatic tissue start becoming cancerous and they become tumors. And so lymphomas are kind of a broad category of some of the lymphatic tissues. So the lymph nodes, the spleen, all start to enlarge, all right? And as they're enlarging, it's because there's a lot of cell division occurring, and that cell division is occurring inappropriately. It's not occurring and enlarging because of an exposure to a pathogen. It's enlarging and occurring because the cells are doing it outside of normal cell cycle regulation. And so this leads to either Hodgkin's disease or non-Hodgkin's disease. And so you might have heard of this. It's fairly common, and Hodgkin's disease is actually pretty treatable form. But again, it's because of cells that live in our lymphatic nodes, so our white blood cells, inappropriately enlarging and going through mitosis. All right, tonsillitis. Many of us have had our tonsils out. You had inflammation of the tonsils. So the tonsils are a lymphatic tissue, and they inflame, and they just don't go down, and then they can start to affect our breathing or our eating purposes, and so we just remove them in many cases with tonsillectomy. Right? And then the last one here, splenomegaly is the enlargement of the spleen. So again, this can happen because you have some blood-borne pathogens in a normal way of the red blood cells and the white blood cells. They hang out in the spleen for a longer period of time. There's lots of white blood cells in there, and they could start to find blood-borne pathogens and remove them, and in doing so, the spleen enlarges. But this is almost, it stays enlarged and it continues to enlarge and it continues to do that inappropriately. And so triggers that can lead that to happen would be scarlet fever, syphilis, and typhoid fever. All right? Now, some immune pathologies can fall into a few different categories. So you can kind of have hypersensitivity, you can have autoimmunity, and, you know, hypersensitivity or hyposensitivity, okay? So hypersensitivity is all of a sudden your white blood cells become excessive and inappropriate to a stimulus. And this is what allergies are. So allergies, is ragweed really going to cause you to get sick? No. But somehow the exposure of ragweed to your body has inappropriately caused white blood cells to act like it's a dangerous pathogen. And so now you have allergies. All right? So you somehow sensitized your body. You made the white blood cells think that pollutant, that ragweed, that mold, that whatever is a deadly pathogen, and your body then from exposure onward always reacts like it's a deadly pathogen and triggers the release of normal inflammatory mechanisms to include histamine and kinins and cytokines and interferons and complements, and causes a huge inflammatory reaction in response to a relatively harmless substance, right? And because it can happen in any place where white blood cells are sitting behind epithelial layers, it can happen in mucous membranes, it can happen on the skin, it can happen in your airways, it can happen in your nose, it can happen a lot of different places, right? Now, another type of immune pathogen that is hypersensitivity would be in your skin. So my kid, you know, this happens a lot in kids. So contact dermatitis. It's kind of a big broad category to say your skin inappropriately is reacting to a chemical that you had on a lotion that it now doesn't like, and so it's making a rash, right? So it's a little bit of a delayed allergic response to chemicals being exposed. And so poison ivy and certain soaps and cosmetics have, you know, soaps and cosmetics have lots of different chemicals in them. And so poison ivy, it's that oil, and it just your skin doesn't like that. But in soaps and cosmetics, your skin doesn't like something in there, and so the exposure of it being in the cream and sitting on your skin for a while then causes white blood cells underneath the epithelial layer of your skin to react, and it causes a rash, okay? And it can lead to a local little skin inflammatory response. All right, the next category are some of our autoimmune diseases. So your white blood cells are potentially acting correctly at some point, but then they start acting against self as well as non-self. So autoimmune diseases means the white blood cells maybe at one point were triggered to turn on and they battled a pathogen and the pathogen got killed and went away. But the white blood cells continue to then attack healthy tissue. And so the response now is they're attacking inappropriately tissues that should be identified as self. So they're no longer immunocompetent. And so some examples of autoimmune diseases are like lupus. It's a systemic problem where white blood cells constantly attack many different cells in the body, and you can develop all sorts of weird symptoms to include a rash, to include kidney problems, blood pressure problems, nervous system problems, skin problems, pain in your joints, because your white blood cells are attacking inappropriately normal healthy cells, but they shouldn't. All right? Transplants fall into here. So you transplant maybe someone's, you know, kidney or someone's heart, and in doing so, now that tissue is not technically you, and so it has some non-self antigens. So in many cases, tissue rejection kind of falls into autoimmune because it's part of you now, even though originally it wasn't. And so this is why matching is so important, because if we can match organs and tissues as efficiently as possible, we decrease the risk that transplants are rejected. And if we can decrease the risk of transplants are rejected, then we have to use less immunosuppression drugs. And if we don't have to use as much immunosuppression drugs, that person doesn't have to then worry about being in a bubble for the rest of their life. They can live a fairly normal life with their new organs. Right? Now, Immunodeficiencies. So this is kind of like a hyposensitivity, or something's not working to the degree it needs to. All right? So immunodeficiencies can happen because the immune system does not effectively defend against a pathogen. And so it can disrupt lymphocyte function, and it can lead to severe kind of long-term pathological chronic conditions and infections, and even lead to cancer being able to take root and then metastasize and survive in your tissue, right? And another type of autoimmune disease is, of course, AIDS, acquired immune deficiency. So you acquire a virus, and the virus is so just smart in its choice of where it's going to target your body that it targets the T helper cells and takes the T helper cell out. And in taking out that T helper cell, you effectively cut off your ability to specifically ever defend against this virus. And you never then are able to generate antibodies against it. You're never able to turn on cytotoxic T cells to kill it. And so that's why it's such an amazing, in some ways, virus, because it developed the ability to find and take residence in a cell that can't really kill and identify itself. So that's why it's so costly, right? Okay. So I've mentioned a lot of pathogens already, and in many ways I take it for granted that you understand what a pathogen is. So very shortly here, and because your book went through this, there are a lot of pathogens. And pathogen is a word to say any organism, anything that toxin that would try to take up residence, cause damage, and use you as a host. All right? And I say it can be a living thing or a non-living thing because viruses are one of those pathogens that technically they're not a cell, so technically they're not alive. But we know cold virus, herpes virus, hepatitis virus, there's lots of viruses out there. And while they don't live maybe long-term on your toilet seat or on your desk or on your doorknobs, if they are contact spread through airway, touching, they can live by moving from cells to cells and moving from host to host, right? Viruses can be treated by antibiotics. It's just we've moved away from treating them from antibiotics, especially to that passive, artificially induced immunity, because they are so fast at evolving and changing their antigen expression that antibodies can stop working after a while. So in Asia right now, there's a form of gonorrhea and there's a form of chlamydia that is, resistant to resistant to antibiotics. So, there's a lot of viruses out there that are becoming like bacteria. They have changed themselves to where giving antibiotics is not being effective anymore. And so instead, your body will make antibiotics, those little Y proteins against the virus. It's just going to take 7 to 10 days in some cases, or even longer. Now, we can make vaccines against some of the viruses out there if the virus is a stable virus. So polio has a vaccine because the polio virus that caused all this paralysis and breathing problems in kids 50 years ago, it's a pretty stable virus. It hasn't changed its structure. It hasn't changed its antigens. So we can make a vaccine against it, and then we've pretty much eradicated, polio from most of the world. Whereas the flu virus, you have to get the flu virus vaccine every year because the flu virus changes just enough every year that last year's vaccine is no longer working. Okay. Sometimes with these vaccines, you have to do multiple injections. So when you get the hepatitis A vaccine, you're targeting the hepatitis A viral strand, and that's a pretty stable virus, but you do it a few times and you do it a few different, sequences to make sure you have enough exposure to enough of the antigen to ensure you actually make your own antibodies against it. Okay? All right. Now, bacteria are cells. So this is a cell, a single-celled organism that is not about going into your cells, it's about taking up residence in and around your cells. So a virus's goal is to go into your cells, to go into, like herpes, to go into your nervous cells, become a part of your neurons, inject their DNA and RNA and become a part of that cell. And viruses are ways we can make our DNA grow longer or add to our DNA. Bacteria, on the other hand, want to hang out in the organs, in the tissues, but not merge and become your cell. Okay, so it's still a separate entity, right? And we tend to, bacteria, while they do change over time, they can evolve, they still are a little more stable than viruses, so that's why vaccines and antibiotics given through a passive mechanism do seem to work a little bit more efficiently than they do against viruses. And there are lots of types of bacteria out there, and that's what micro is about, learning about bacteria and viruses and their structure and features and how to test for them, right? So all of our staph infections, all of our strep throat infections, all of our, there's a form of bacterial meningitis, there's a form of viral meningitis, pneumonia, that's why people get the pneumonia vaccine, because it's a protection against a bacteria that takes up residence and causes problems with your lungs to fill with water, breathing issues. Tuberculosis, it's a big bacteria that likes to tear away your lung tissue and make you cough up blood. Tick-borne bacterial disease like Lyme disease, E. coli, chlamydia, gonorrhea, syphilis. Syphilis is sometimes known as the French whore disease because it was common in French brothels back in the 17 and 1800s, right? And chlamydia and gonorrhea, there are forms of this in Asia, so if you travel to Thailand and you want to go to the Kuchiku shows or whatever, just note that many of those brothels there could have girls that have a non-bacteria responding form of chlamydia and gonorrhea. Okay. All right. Another type of pathogen is a fungus. So remember, fungus is a different kingdom. You know, so we usually think of mushrooms when we think of fungus, but yeast. Yeast is a fungus. And yeast is located on our bodies, and we find that when it grows inappropriately, we can get jock itch, we can get ringworm, and we can get yeast infections in our vaginal regions or our armpits. And that, again, is because we usually take antibiotics and kill a lot of good bacteria, and so some of the yeast and some of the fungus can grow there, right? And you can get to all types of fungal infections through barefoot exposure to bathrooms and any place that mold can grow. In fact, we have mold in our building here through the air vents. So every time you breathe in the Meramec buildings, you might be getting exposed to a mold and a fungus that can make a fungal respiratory infection, potentially. Okay? So there was not too long ago, there was some contamination of mold at a pharmacology lab somewhere in the middle of the world, in the middle of the country. And all these people that were getting, I think it was insulin, so you know with the diabetes out there, these people were getting insulin and they were injecting themselves with their insulin, not knowing that they were also injecting themselves with fungus that had lived in the bottle, inside the bottle with the insulin. And all of a sudden, all this fungal meningitis, which is very rare, started happening. And I mean, it was killing people. And they leaked it back to everyone was getting their drugs from a certain pharmacy, and the pharmacy had contamination of some of their sterilization equipment with mold, and every time they went to sterilize glass bottles, those little bottles that people pulled drugs out of, they were spreading mold, fungus that could kill people with attacking their brain and spinal cord, right? All right, a little less common because of our hygiene and because of our practices of, you know, sewage control are protozoans and parasitic worms. So if you read any of like World War II and the Holocaust camps and the POW, because I read The Summer Unbroken and I've read some other books, the protozoans that can form and cause giardia, basically it happens, again, with all they had to sleep and drink and eat food that they used their own poop to fertilize, and they had very bad sewage, and so it was very common, again, before the common now sewage pipes and water treatment that we do. Malaria comes from a mosquito, and it gets into your red blood cells, and it's a little parasite, and it lives in your body. And depending on the conditions of your body, you'll have little flare-ups of when the protozoa in the organism is what do you call it? When the organism is reproducing, multiplying, and trying to take over more red blood cells, and then you'll have periods where you're not suffering from it, okay? Everybody's heard of nits and crabs and lice, and you hear about kids all the time, you know, there's a nit and a lice outbreak. Those are parasites. Granted, big scheme of things, not a huge deal, but like tapeworms, if they get into your system, you ingest them, they could potentially, you know, cause problems. And tapeworms will take in residence in your small intestines and then steal your food, okay? All right, the heart, the cardiovascular system is now we're moving on. So that was the lymphatics. So the heart is going to be a big component of our cardiovascular system. Our cardiovascular system is the heart, the blood, and the pipes. The heart gets a lot of attention because it's an extremely important organ. If it ever stops working, your blood stops flowing, oxygen delivery stops, glucose delivery stops, waste pickup stops of carbon dioxide and other things to remove at the kidneys. Big picture, the heart has 4 chambers, 2 atria that sit near the base or the top of the heart, and then 2 bigger chambers known as the ventricles. The right ventricle gets blood from the left atria. The left ventricle gets blood from the left atria. Right blood is pumped to the lungs. Left blood is pumped to the body. So the right atria, right ventricle to the lungs is known as your pulmonary circuit, and your left atria, left ventricle to the aorta and all your systemic arteries is known as your systemic circuit. All right? The heart is fed by special capillaries and vessels that come from the aorta. All right, they kind of leak blood into these little vessels known as your coronary vessels. And they feed the cells that live and are part of the heart organ itself. So all these cells in the middle. The outermost part of the heart is known as the epicardium. The innermost is the endocardium. The muscle is in the middle. Okay? And I threw up a lot of the terms here, or just giving you your bearings of what are some of the important vessels that attach to the heart and take blood to and away from the chambers of the heart. Okay? Now the conduction system of the heart is going to be how the heart knows when to beat. And it knows when to beat intrinsically, meaning there are certain places and pockets of cells in the heart that will spontaneously undergo a depolarization, a change in electrical activity, and it leads to contraction of the heart tissue. And that main spot is known as the SA node. Right? And the SA node is known as your pacemaker because it's the place in the heart where your cells are able to reach depolarization, reach the changes of the electrical properties of the heart that lead to contraction. And when it triggers and starts to depolarize and starts to change the electrical activities, that will spread first through these internodal pathways, through the atrias, to this point here where the right and left atria and right and left ventricle all come together, known as your second node, your AV node, your atrioventricular node. Okay? And this node is important because it does 2 things. It slows down and gives you a little bit of a delay. Now, by giving you a little bit of delay, the atrias will contract before the ventricles, and therefore you end up with atrial systole, atrial contraction, blood flowing into the ventricles, topping off and filling before the ventricles are contracting. Right? Now, the next part of the AV node is you have to get the electricity to the tip and to the apex and then up and around to all of those cells around your ventricles fast. And so this is where the AV bundle, the bundle branches, and the Purkinje fibers allow you to get energy to the tip and then around. So when the ventricles depolarize and contract, they depolarize from the apex, the tip of the heart, upward, and therefore how does blood flow? It flows up into the major vessels on the right side, the pulmonary vessels, and on the aortic, the left side, the aorta, and to all the systemic vessels. How do we see if the heart's doing the right electrical properties? We do EKGs, also known as ECGs. And so what ECGs are, they're a measurement of the electrical changes occurring in the heart. Right? And so in a lead 2, so this is normally a lead 2 handout, which is measuring the electrical changes from the right arm to the left leg in a negative to positive direction, we are going to see anytime that the depolarization occurs, we're going to see usually this little upward deflection. Right? And so the P wave represents the first wave when the right and left atria is depolarized, and then they're going to contract. The delay is important because we get the ventricles to fill with blood. When the ventricles depolarize, it's very fast, so it makes a long or high amplitude, and it's going to be the QRS complex. And it's going to be very fast because they're all depolarizing at the same event, and then following that QRS complex, we're going to get the ventricles contracting. Now, at some point, the ventricles are going to repolarize, and they're going to repolarize in the opposite direction that they depolarized. And because they're going to depolarize from the tip towards the right arm, they're going to depolarize from negative to positive in the opposite direction that the reading is taken, and so that's why the repolarization of the ventricles shows the second blip, the little T wave. Okay? And so when we do EKGs, we're basically seeing, are you generating a P wave? So is your SA node in charge? For every time your SA node generates a depolarization and an atria contracts, are you getting the AV node to then transmit that next signal into the ventricles? So does every P wave follow a QR or come before a QRS complex? And then do you have a QRS complex that's strong, swift, and quick? And then do you get repolarization of the ventricle altogether as well? And if any of those things are off, then you know something is off with the electrical properties and even therefore the contractile ability of the organ of the heart. Okay? And so when we start looking at heart function, we want to look at cardiac output. How much is the heart beating in a given time? And then how much blood is it pumping out every beat? So cardiac output is usually for a minute in time. You should pump enough blood to have 5 liters of blood circulating through your body every minute. So normally you'll be somewhere around 60 or maybe 80 beats a minute, and your stroke volume will be somewhere around 80 milliliters of blood. And so if you multiply these two things out, right, you should end up with, if it's 60 times 80, you end up with 48,000 milliliters of blood a minute. And so because it's biology, we just round that up to about 5 liters of blood a minute. Okay? When you exercise, cardiac output will go up to as high as 16 liters a minute. So you're usually maybe exercising at 160 beats a minute, and maybe your stroke volume is now 100 milliliters, and so that gets you up to 16 liters a minute. Okay? So when we look at heart function, we want to know that your heart is pumping a normal amount a minute, and it's pumping out a normal volume of blood a minute. And so anything that changes related to how much volume you leave the heart per beat and how many beats per minute it's taking or per hour is going to then affect heart function and whole body function. All right? You can change heart rate by changing hormones like epinephrine or thyroid hormones. So someone who has hyperthyroid might have a really erratic high heartbeat. You can change the heart rate with autonomic. So when you are scared and you have output of sympathetics, norepinephrine, adrenaline, that's going to make your heart go up, versus when you're sleeping and you're eating and you're relaxing, you have acetylcholine, and so your heart rate's down. On the other side, the stroke volume, it's all about getting more blood to start in the ventricle before they contract. So at the end of the ventricle relaxing, end of ventricle diastolic periods, you have a high amount of blood in there. And then when the end of the ventricle contracting, the end of its systole, you have a low. So stroke volume is the difference of what you start with in diastolic volume minus what you end with in systolic volume. So anything that usually makes this go up makes cardiac output and stroke volume go up. Anything that makes this go down, start with less blood in the heart, you end up with less stroke volume, less cardiac output. Anything that makes your heart beat better and squeeze more blood out and makes in systolic stroke volume go down, so there's less blood left is going to make stroke volume go up. Anything that makes more blood stay in the heart, and therefore in systolic volume go up, is going to decrease stroke volume. Okay? So lots of cardiovascular pathologies. I just picked a sampling. Hypertension is a pathology related to afterload, meaning because of the vessels having a lot of resistance, a lot of force you have to push against, that can lead to damage to those vessels, which is going to be a problem, and it can lead to damage in the heart and damage to the heart's ability to pump blood, a sufficient stroke volume out, and enough pressure to get blood to the different areas of your body. All right, hypertension can result because you get atherosclerosis, a thickening or hardening of the arterial walls, and then atherosclerosis can cause heart problems when it happens in the coronary system because it can start leading to death of heart cells. So lots of different reasons why hardening of your arteries is a bad thing. You can have valve disorders, so anything that messes with your atrial ventricular valves and lets blood go back to the atria when it should stay in the ventricles, or any valves like the semilunar that lets blood fall back in the ventricles instead of going to the vessels, or if those valves get tough and they don't let the blood flow into the ventricles or out of the ventricles, all of that's going to play a role in decreasing potentially stroke volume and decreasing your heart's effectiveness. Inflammation. You have connective tissue in the heart, therefore you have white blood cells. White blood cells can cause inflammatory responses. So if you get inflammation of the outer covering of the heart, inflammation of the connective tissue in the heart, you can have endocarditis or pericarditis. Okay? Arrhythmias, anything that messes with the heart rate, the hormones and the autonomic output that mess with the SA node, mess with the AV node's ability to conduct or respond or depolarize. That can lead to arrhythmias. And arrhythmias can lead to the heart cells not being in sync, and that can lead to fibrillation. All right? And that's what you learn in CPR. And then eventually the heart's just not an effective pump. It loses cells, or the cells are not strong enough, and it doesn't have enough cells to then effectively push on the chambers. And that is when it's insufficiently pushing out cardiac output, it's in heart failure. And we can treat that with drugs, and that can happen for a variety of reasons, but eventually this can lead to death. We can keep you alive in heart failure, but you're not going to run marathons. And we can potentially transplant, but your new heart will probably start to fail eventually too. The last bit was, what are some of the things in the book we can do to prevent cardiovascular disease? And this is everything you should learn in a health class. Watch your diet. Don't be overweight. Make sure you watch your exposure to saturated fats. That damages cells of the heart, damages cells in the vessels. Don't overstress your heart by increasing resistance, afterload, making yourself bigger for body weight and composition, more fat mass. Exercise, challenge the heart to be better responders to neuro responses, hormonal responses, and to do a better job of being stretched and pressed on the vessels and the walls. All right. Avoid tobacco and the exposure to carcinogens. Manage your stress and avoid becoming diabetic and high blood pressure. So manage your blood sugars and your blood pressure. All right, and that is it for today, this morning. I will do the next lecture at 2:45-ish when I get done with lab. All right, if you have questions, use the forums, and good luck. Remember, you have a quiz due Sunday, 11:59 p.m. over this material plus the material I'll lecture on later this afternoon.